4.2 Diffusion and Facilitated Diffusion. Having looked at the structure of the membrane and looked at some of its function, we need to look a little bit more closely now at exactly how things cross the plasma membrane to get into and out of the cell. The plasma membrane is permeable. Permeable means that things can cross through. Specifically, the plasma membrane is called differentially permeable or selectively permeable. And what this means is only very specific types of molecules can cross the plasma membrane. We can broadly classify types of movement across the plasma membrane as either passive transport or active transport. The difference, passive transport does not require that the cell expend energy. Active transport does require cellular energy, usually in the form of ATP. We're going to look at several types of passive transport, these that do not require cellular energy. The first one is diffusion, and then facilitated transport. And thirdly, osmosis. And then we'll look at some active transport pumps, and then we'll end with some vesicular transport. Diffusion is the net movement of molecules from an area of high concentration to an area of low concentration. Things move down their concentration gradient. So movement is from high concentration down to low concentration. Whenever things are diffusing in a cell, this does not require that the cell spend any energy. The movement is due to just random vibrations and collisions. As molecules move, vibrate, they bounce into one another, they bounce into other things in their environment and deflect. And this random movement causes the molecules to spread out. And so the net change is always going to be movement from high concentration into low concentration. This is just simple diffusion. Here's a simple example. If we take a molecule of dye and we drop this in a beaker of water, we have an area of high dye concentration in the crystal, and over time, those dye molecules will begin to move out. And so they will spread out in the water, moving from that initial high concentration out into surrounding low concentration, simple diffusion. And this will continue until eventually we have equal distribution of the molecules. We would call this equilibrium. Now, this doesn't mean that movement has stopped. Certainly, all of the molecules are still moving within this beaker, but after we reach equilibrium, there is no net change in concentration. We are going to remain relatively evenly distributed throughout the beaker. Anything can diffuse. If you open a bottle of perfume and wait just a moment, then within just a few seconds, you can smell the perfume. A simple example of diffusion. Those perfume molecules are highly concentrated inside the bottle. Whenever you release the cap, those molecules can begin to spread out. Many things diffuse in the body. Whenever we inhale, oxygen diffuses from the lungs into the blood. It's moving down its concentration gradient. Simultaneously, carbon dioxide, which is a waste product of cellular respiration, is going to diffuse from the blood into the lungs. So in both cases, these molecules are moving down their uh, concentration gradient. There are several factors that can influence the rate of diffusion, how quickly diffusion takes place. One of those is the size of the concentration gradient. If you have an area of very high concentration immediately adjacent to an area of very low concentration, we would say that's a steep gradient and diffusion would take place more rapidly. Also, the size of the diffusing molecule influences the rate. Generally, large molecules are going to move more slowly than small molecules. Temperature, uh, all molecular movement increases as temperature increases. So certainly an increase in temperature would increase the rate of diffusion. And also the type of medium. Gases are going to diffuse more rapidly because these are small molecules. It's a less dense medium. Things will diffuse into water or a liquid a little bit more slowly, and diffusion through a semi-solid medium is going to be slower yet. And so the type of medium will influence diffusion rate. Now we have to think just a little bit. What types of molecules can freely diffuse across the phospholipid bilayer of the plasma membrane? So we have to remember the structure of that bilayer and the nature of the phospholipid and the phospholipid bilayer. Generally, small, nonpolar, uncharged molecules can freely diffuse across the lipid bilayer. So if we have a concentration difference, a concentration gradient on opposite sides of the phospholipid bilayer, these small, uncharged, nonpolar molecules can freely cross the membrane. So here's a diagram. Certainly a large molecule, large macromolecules, such as proteins, large carbohydrates, even simple sugars, monosaccharides, are just simply too large to pass between the phospholipids. And so large molecules are blocked. They cannot freely diffuse across the membrane. Charged molecules or ions or very highly polar molecules. These types of molecules, like water, they're very hydrophilic. And so they are going to be repelled by that hydrophobic core of the phospholipid bilayer. So very polar or charged molecules cannot cross. Generally, small, uncharged, lipid-like molecules can freely pass through the hydrophobic core, and these small molecules can cross the plasma membrane. Now, one exception to this is water. Water certainly is polar, but it's also a very small molecule, and there are some pores that do allow water to cross from one side of the plasma membrane to the other. But again, generally, it's only small, uncharged, nonpolar molecules that can freely cross through the phospholipid bilayer. Another type of passive transport is facilitated transport, sometimes called facilitated diffusion. I prefer to call it facilitated diffusion. This is the movement of molecules that are, in fact, charged or highly polar, or in some cases just too big to cross the phospholipid bilayer. And many times we still need to get these molecules into or out of the cell, but these can't wiggle through those phospholipids. And so we have to have another method of getting these molecules across the membrane. This is still movement down a concentration gradient, which is why I like to call this facilitated diffusion. We're still going down a concentration gradient. It's facilitated, however, because we're going to have to use some of those protein carriers that we identified earlier in this lesson. So here is an example. You can see the phospholipids illustrated here in blue, phospholipid bilayer. There's one of the carrier proteins, one of the integral membrane proteins, and now we have a large concentration on one side of the membrane of a large molecule, and we need to get this large or possibly very polar molecule into the cell. It can't go through the phospholipids, and so this carrier protein can bind this molecule and carry it across the membrane. This is still a passive mechanism, the cell's not having to use any energy because this is driven by the concentration gradient.